Hi guys, welcome back. Lesson 8 on the musculoskeletal system. So we're going to be looking at the skeleton and the muscles, and you should, as always, have a knowledge of this from level 2 that we're going to be looking to expand on today. And in particular, we're going to be looking at the cellular structure of muscle fibers, so exactly what they look like at a cellular level. And at that cellular level, so inside those muscle cells, there's something called sliding filament theory, which you're going to be able to describe. You're going to be able to explain the effects of different types of exercise on muscle fiber type, and you're also going to be able to identify and locate the muscle attachment sites for the major muscles of the body. So this is quite a big one. There is a lot to pay attention on. As always, as we always say, benefit of the online training is if you don't understand anything, please play it back, have another listen, make sure you understand and make sure you can get those marks when it comes to exam time. So, first thing that we're going to look at is this structure of a muscle, and this should be looking really quite familiar from the work that you did at level 2. We've got the tendon at the end and then the fascia from there, and then we've got all of these other bits that are not labelled yet but they soon will be, and you need to know exactly what they all are and exactly what they all do. But in simple terms, this is a skeletal muscle, and it's made up of lots and lots and lots of rod-like structures, and they're called muscle fibers. And all of these muscle fibers, they run parallel along the length of the muscle. And if we look a little bit closer, we'll be able to see each series of fibers themselves and what they're made up of. But what we have is surrounding and protecting all of the muscles, there are sort of collagen-based membranes, and the outermost collagen-based membrane is called the epimysium, and essentially that is a sheet of elastic tissue that wraps around the muscle, keeps it in place, keeps it nice and tight, and makes sure that everything is where it's supposed to be. And within the epimysium, there are groups of muscle fibers, and these are called fascicles, alright, or fasciolae. But essentially what they are, a fasciole or a fascicle, is a group of muscle fibers, and each fascicle has its own sort of protective sheath around it, and this is called the perimysium. And peri means around, so it's around the fascicle. Alright, that's the perimysium that goes around the fascicle. Alright, within each of the fascicles there are bundles of muscle fibers, and they're separated from each other by something called the endomysium. So as you can see, if we look a bit closely at this one, lots and lots of circles. All of those circles are endomysiums, and inside the endomysiums we have the individual muscle fibers, and the proper name for an individual muscle fiber is a myofibril. So hope that's refreshed you a little bit. Should all make sense. You should know what each bit of connective tissue is and where it comes in, and what each group of muscle cells is also called. If you don't know, have a little look, play it back, have yourself a little bit of revision, because that will no doubt come upon the exam, and there'll be valuable marks that you can pick up on there. However, at level 3, we are interested in what's going on inside the myofibril. So at a real cellular level, so inside those cells, what is happening? And at the top we have the individual myofibril, and the individual myofibril is broken down into individual segments, and these are called sarcomeres, as you can see. Alright, so there are 1, 2, 3, 4, 5 sarcomeres making up that there myofibril. If we looked inside a sarcomere, this is what it looks like. Lots and lots of series of this. So big fat sort of pinky lines, smaller, thinner, purple lines. And the big fat ones, they are something called myosin. They are the thickest protein filament. And then you have actin, which is the thinner protein filament. So inside the sarcomere, we have myosin, which is the thick one. We also have actin, which is the thin one. All right, and what we want to understand and what we want to know more about is the interaction between the myosin and the actin, because when these two filaments interact with each other, they cause muscles to contract. And we're going to look at it in more detail, but essentially what's going to happen is the actin is going to slide across the top of the myosin. That is why it's called sliding filament theory. This may help you a little bit more. So, here we have a sarcomere. You can see where you've got the little zigzaggy lines. That's the end of each sarcomere. And inside, again, we have the big thick one, which is the myosin, and the smaller little one, which is the actin. And same thing here, broken down at the bottom. And when movement happens, what happens is the myosin heads, so these little head-like bits down on the red bit there at the bottom where the cursor is, alright? Those little head-like bits, they'll reach across, grab on to the actin, and they'll rotate and slide along. So, you imagine they keep doing that until the actin, as you can see here, reaches up, rotates, and pulls it along. It's the myosin reaching out to the actin, twisting, and pulling it down. However, we need to know exactly what's going on, sort of scientifically, for that process to happen. And what you should remember, back from level 2 when we looked at how we create energy, is something called ATP. Essentially, ATP is what humans use to power their bodies. And what happens is when we create ATP, which we'll look at that in a bit more detail later on, but let's just for now, let's just assume we've created some ATP. Don't worry about how that happened. We create some ATP, and the ATP primes the myosin for activity. So it gets it ready for activity, gets it ready to go. All right? And what happens is it binds, so the ATP molecule, all right, binds with the myosin head. And then energy from the ATP is almost immediately transferred to the head, a bit like cocking a gun. So when you pull back when you cock the gun and get it ready to go, that's what it's a bit like when the ATP joins with the myosin. It's just ready to go. It's set up, getting ready to pull the trigger. However, the myosin can't join up with the actin and rotate round and cause that movement, cause that slide, unless there is something called calcium present. All right, because in a relaxed state, the sites where the myosin binds onto the actin, all right, those sites there, they're blocked. They're blocked by a combination of other molecules, but the myosin can't join on unless we do something, essentially release calcium to get rid of those particles on there that are stopping it from joining up and stopping it from sliding along and stopping movement from happening. So surrounding the myofibrils, there's a network of tubes which are called the sarcoplasmic reticulum. All right, and essentially, what the sarcoplasmic reticulum is a series of calcium reservoirs. So when these reservoirs are stimulated, and we'll look at how that happens in a minute, but when the reservoirs are stimulated, they release calcium. The calcium fluid surrounds the myosin and the actin, and it gets rid of the blocking agent or blocking molecule, therefore leaving the myosin to bind onto the actin, and movement can therefore happen. So what we can conclude from that is, without the influx of the calcium into the muscle fibre, sliding filament theory cannot take place. So that then poses the question of what happens for this calcium to get released. And what happens is, so the stimulus for the calcium to be released is the spread of electrical activity, or what we may know from level 2 as an action potential. All right, the spread of electricity along the length of the muscles in a resting state, okay, means that that is an action potential. That spread of electricity along the length of the muscles. And what happens is that in a resting state, muscle membranes are negatively charged on the interior and positively charged on the exterior. A little bit like a battery has stored electricity. It's the same sort of principle. And the difference between the two gives us potential for energy. And it's a bit like having that battery, having that stored electric in there ready to go. That's what that positive and negative do. Just think of it as having stored energy ready to go. But that energy can't be released unless we have the presence of calcium and ATP to make that happen. Okay? And the arrival of an action potential at a neuromuscular junction, so that's where the nerves meet the muscle. So the arrival of electricity, where the nerves meet the muscle, causes the release of a neurotransmitter, which in turn then causes sodium to rush through the muscle membrane, and as a result, there's a reversal of electricity, which, if great enough, will cascade through the muscle fibre as action potential, thereby triggering the calcium release. So it's that where the nerves meet the muscle, the electricity travels down there, and if it's a strong enough signal, the electricity is strong enough, it will release sodium, which will then, as a result, incur the release of the calcium, which will get rid of the blocking agent and allow the myosin to bind with the actin, and movement will happen. One more time we'll look through this just in case you didn't get it. Myosin reaching up with the presence of calcium, grabbing onto the actin and sliding. Okay? So, that was my go at explaining it. What I also want to do is have a look at this video because I think this is very, very valuable. So,
Romeo and Juliet, Helen and Paris, Tristan and Isolde. These famous star-crossed lovers bring to mind insatiable longing, forbidden love, and tragic separation, and poets and emo rockers love them for it. But you know where else you can find a nice, hot romance? Your muscle cells. They've got their own famous coupling, a pretty pair of tiny protein strands called actin and myosin. Romeo and Juliet may have set a chain of tragic events into motion with their infatuation, but deep down in the cells of your muscles, the hot protein action between actin and myosin is actually literally causing all of your motions. All of them. And I don't just mean voluntary stuff like walking down the street or moving your mouth so you can talk or chew chips, because your muscles also support your weight and help fend off gravity. The amazing thing about your complicated, self-healing, blood-guzzling muscle tissues is that they turn chemical potential energy into mechanical energy, or movement, simply by doing two things: contracting and relaxing. And that contracting and relaxing is exactly what's fueled by the constant coupling and separation of biology's greatest lovers. Somebody get these proteins a movie contract. You will recall from our early lessons on tissues that you're kept alive and moving by three types of muscle tissue: smooth, cardiac, and skeletal. Your smooth muscle tissue is found in the walls of all your hollow visceral organs like your stomach and airways and blood vessels, where it involuntarily and very usefully pushes fluid and other material around by contracting and relaxing over and over. Your heart is so important that it gets its very own muscle tissue type, cardiac muscle, which looks striped or striated. It also functions involuntarily to keep your blood pumping without you having to think about it. But when you hear the word muscle, you're probably thinking of the kind that you see on Chris Evans when he first walks out of that machine in Captain America. And those types, the ones you can see and feel and flex, are your 640 skeletal muscles. They're striated like cardiac muscle tissue, but they're also mostly voluntary, meaning you have to think about using them and activate them with your somatic nervous system. Most of them attach to your skeleton and create movement by pulling bones in different directions as they contract. Each one of your different skeletal muscles, like your biceps brachii or vastus lateralis or gluteus maximus, is technically its own organ made up mostly of muscle tissue, but also of connective tissue, blood vessels, and nerve fibers. And because your muscles are voracious energy hogs, dogs. Each one is rigged up with its own personal nerve to stimulate contraction, and its own artery and vein to keep it well fed with all the blood and oxygen and nutrients it needs to operate. But to understand those operations, we first need to get a grip on the anatomy of a skeletal muscle, which involves fibers within fibers and lots of layers. Basically, a skeletal muscle is constructed like a really sturdy piece of rope. Thousands of tiny parallel threads called myofibrils squished together to form muscle fibers, which are your actual muscle cells with mitochondria, multiple nuclei, and a cellular membrane called a sarcolemma. Those muscle fibers then form larger string-like bundles called fascicles, which combine to form the larger rope-like muscle organ like your biceps brachii. Overall, this bundles of bundles configuration makes muscle tissue fairly sturdy, but considering how much abuse your muscles take when you do something like pretty simple, like lift a big bag of dog food, it's no surprise you need a little help. That's why every muscle contains a few different kinds of supportive sheaths of connective tissue, the protective reinforcements to keep that bulging muscle from bursting. So that is the structure part of the story. But if you want to get into the nitty-gritty, the down and dirty of how you actually move, well, there are rules. Really just two main rules, and they have to do with proteins. And they're both true for a lot of the proteins we talk about, whether they're enzymes or ion channels or receptors or muscle proteins. And these rules are: 1. Proteins like to change shape when stuff binds to them, and 2. Changing shapes can allow proteins to bind or unbind with other stuff. So keep those rules in mind while we see how a muscle fiber contracts and relaxes. Now, remember those tiny myofibrils that bundle up to form your muscle fibers? They're divided lengthwise into segments called sarcomeres, which contain 2 even tinier strands of protein, 2 different kinds of myofilaments called actin and myosin. And it's their angsty story of star-crossed love that fuels every movement your body could possibly dream up. A sarcomere contains both thin filaments, made up mostly of 2 light and twisty actin strands, and thick filaments composed of thicker, lumpy-looking myosin strands. Each sarcomere is separated by what's known as a Z-line at either end, which is just a border formed by alternating thin filaments in a kind of zigzag pattern. A muscle contracting is all about sarcomeres contracting, bringing those Z-lines closer together. Alright, so now comes the romance. When your muscle cells are at rest, your actin and myosin strands don't touch, but they really want to. Specifically, that club-headed myosin wants to get all up close and personal with the actin. When this happens, and it will eventually, it's called the sliding filament model of muscle contraction. But in the meantime, like in any good love story, the pair have some obstacles to overcome. Namely, actin is blocked by a couple of protein bodyguards called tropomyosin and troponin, which keep getting in the way. Luckily, these guards can be bought off with a little ATP and some calcium. I prefer cash and nachos, but whatever. Remember, ATP is kind of like molecular currency. It contains chemical energy, and your muscles are all about converting chemical energy to motion, so they're always hungry for more ATP. Your muscle cells have lots of nuclei, but some of them also have a lot of mitochondria, whose sole purpose in life is to crank out ATP. And muscle cells also have their own version of an endoplasmic reticulum, the cell's transport and storage system, but in this case it's specialized, so it gets a special name, the sarcoplasmic reticulum. Its walls are loaded with calcium pumps, which use ATP to save up a bunch of calcium ions. And it's also studded with calcium channels that are linked to voltage-sensitive proteins in the membrane of the muscle cell. Say I want to move my arm. My brain sends an action potential along the motor neuron until it synapses with a muscle cell in my arm. The receptors on that muscle cell are ligand-gated sodium channels. So when the motor neuron releases our old friend acetylcholine into the synapse, the channels open up and create a rush of sodium into the cell as a graded potential, which, if it's strong enough, causes nearby voltage-gated sodium channels to open. Now, I want to take a second and point out here that we're still talking about an action potential, but not in a neuron. This is happening in a muscle cell, people. So that action potential zips along a muscle cell's membrane, the sarcolemma, which has lots of tubes that run deep inside the cell, called T-tubules. When the action potential travels down one of those tubes, it eventually triggers the voltage-sensitive proteins that are linked to those calcium channels in the cell's sarcoplasmic reticulum. When those channels are thrown open, the calcium stored inside rushes into the rest of the cell, and finally myosin is like, yes, here we go! At this point, the myosin is totally stoked, because the bodyguards that have been frustrating it are in for a big, irresistible distraction. That's because the protein troponin just loves to bind with calcium. And remember, when stuff binds to proteins, the proteins change shape. So the calcium latches onto the troponin and causes it to pull the other bodyguard protein, the tropomyosin, away from the sites on the actin strands that the myosin really wants to get its paws on. And suddenly it's all, okay. But the only myosin heads that can bind to those newly exposed sites are the ones that are ready for action. That is, the ones that have already grabbed a molecule of ATP that's been floating around and broken it down into ADP and the leftover phosphate. When a myosin head does that, it moves into an extended position, kind of like a stretched spring, still holding on to the ADP and the phosphate, and still storing the energy that was released when they were broken apart. So after all that, with the myosin primed for action and the bodyguards out of the way, the myosin finally binds to actin, and it is beautiful. When they bind, the myosin releases all that stored energy, and in the excitement of it all, the myosin changes shape. It pulls on its precious actin strand, kind of like pulling a rope hand over fist. In the process, it shrinks the whole sarcomere and contracts the muscle. That's the sliding part of the sliding filament model. Now, with its energy spent, that little head has no use for the ADP and the phosphate, so they unbind with it because, remember rule number 2, changing shape encourages proteins to bind or unbind with stuff. That unbinding causes a small change in its shape, which lets a fresh ATP bind there in its place. That binding causes another shape change, but this time it causes the myosin to release from the actin in a tear-jerking scene like some microscopic recreation of the finale from Titanic. But fear not, this epic is not quite over, because this is when the myosin breaks down its new molecule of ATP into ADP and a phosphate, which moves it into the armed position yet again, getting it ready for its next rendezvous. And meanwhile, those calcium pumps are working hard to restock the calcium in the sarcoplasmic reticulum, so they start grabbing the calcium that's floating around, causing calcium to unbind from the troponine. When it unbinds, the resulting shape change puts the tropomyosin bodyguards back into place. It's a circle. Or potentially a big Hollywood franchise with lots and lots of sequels. It keeps repeating itself many, many times every moment while I sit here and talk, and while you sit there and eat and text and take notes, the whole drama replaying itself over and over. Kinda like you'll have to play this video over and over again to get all the little steps of the sliding filament model straightened out. But hey, some stories get better the more you hear them. If you do watch this one again, you will relearn that your smooth cardiac and skeletal muscles create movement by contracting and releasing in a process called the sliding filament model. You'd also relearn that your skeletal muscles are constructed like a rope made of bundles of protein fibers, and that the smallest strands are your actin and myosin myofilaments. It's their use of calcium and ATP that causes the binding and unbinding that makes sarcomeres contract and relax. Special thanks to our headmaster of learning, Thomas Frank, for his support of Crash Course and free education. And thank you to all of our Patreon patrons who help make Crash Course possible through their monthly contributions. If you like Crash Course and want to help us keep making great new videos like this one, you can check out patreon.com/crashcourse. Crash Course is filmed in the Dr. Cheryl C. Kinney Crash Course studio. This episode was written by Kathleen.
Okay, so we'll leave that there. Hope you found it very helpful. As always, you can play it back if you need to, but I'm going to exit this now. I'm going to jump back into our presentation if I can find it. And what you can also do, guys, if you want to get some more information on this, have a look at the manual. If you find it easier to understand rather than watching video and hearing someone speak, also have a look at the manual, because pages 28 to 30 will also be able to help you out with sliding filament theory. But what I want to look at now is fast and slow twitch muscle fibers, and sort of the main point that I'm looking to get across, and the main thing that you probably need to take away, is that whilst it's acknowledged that exercise in general will have a positive impact on the sort of development of muscle tissue, there are certain types of activity that have been shown to influence different types of muscle fibers. So, sort of to put it a bit more simply, type 1 or slow twitch, type 2A, and type 2B or fast twitch. Those are the names of the different fibers. So we have type 1, we have type 2A, and we have type 2B. And type 1 are our slow fibers. So those are the ones that we use when we're doing our long distance type activities, sort of running a marathon, long distance cycle, continued long use of the muscle. All right, those are our type 1. And they are called slow twitch muscle fibers. Then we have our type 2, which are fast twitch muscle fibers, and they are the muscle fibers that we use when we're going to do sort of a sprint type activity, or something that is very anaerobic in nature with reduced blood supply in comparison to the type 1. But the I suppose where we have a bit of an area for, not an area for concern, but an interesting area, is that the type 2A fibres, they have all the characteristics that we find in other fast twitch muscle fibres, but they adapt a little more and they can take on the properties of a type 1 fibre, dependent on the activity that that you're doing. Essentially, if you do more long distance work, more cardio work, the type 2A fibres will become a bit more like type 1 fibres. If you're doing more weightlifting, if you're doing more sprinting type work, the type A are going to come across as being more like type 2B. And you also need to be aware of the little bullet points below. So it makes sense when you think about it. The ones that we're using all the time for long distance stuff, they have rich blood supply. Alright, they're red because they've got more blood in them and they have a rich blood supply. They have lots of oxygen going to them, aerobic supply. They're rich in mitochondria, so they're able to produce more and do more work. They don't produce quite as much force, and they're slower to fatigue. And essentially, the type 2B, they're the opposite. So they have a poor blood supply, and they're white. They're anaerobic. They have poor mitochondria, so they can't do lots of work, and they can't create lots of ATP. And they have higher force production, and they're quicker to get tired. So that's the difference. And you also have a little bit in the middle where essentially where those fibres can take on aspects of the other two. So whether it be type 1 or type 2B, in the middle can take on aspects of both. The last thing we need to look at is we need to look at origins and insertions, okay? And for this is going to be a bit of learning on your part because we have the anterior muscles and we also have the posterior muscles. So anterior means front, posterior means back. And sort of pretty much all skeletal muscle is attached to bone through the tendons, which we already know. And when the muscles contract, they exert force on the bones, which help to support and move our bodies. And in most cases, one end of the muscle is in a fixed position, while the other end moves during contraction. And it's the origin is the attachment site that doesn't move, and it's the insertion, which is the bit where we have the movement. So what you need to do, guys, you need to go to page 36 of your manual. When you're there, there's a big chart and it shows you all of the origins and the insertions. You need to have a look through that, and you need to be able to you need to know where all the different muscles originate from and where they insert into. And having that knowledge, because this always comes up on the exam, okay, so make sure you're on page 36 and make sure you're getting that knowledge down. There's not too much point in having a PowerPoint on it because it is a case of looking at name of the muscle, where it starts and where it finishes, okay? And it's remembering that. So, it's been an interesting one. There's been a lot of knowledge to take in on this one. So, take your time, make some notes, have a browse through the manual and reinforce that learning if you need to. But make sure that you can do all of these bullet points on here. So, explaining the cellular structure of the muscle fibres, describing sliding filament theory, and being able to explain the effects of different type of exercise on muscle fibre, and being able to locate the muscle attachment sites, all right? Make sure you can do that because you're going to need to be able to do it so that you can essentially pass the exam, because these areas do always come up. Thanks for listening, and I'll see you again when we do part 2 on the muscular skeletal system.